ஹாய் காய்ஸ், வெல்கம் டு லேர்ன் வித் கோக்ஸ். இன்றைக்கி வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி ஒரு ஆட் பார்த்துடலாம். BuyMote ஷாப்பிங் ஆப். இது ஒரு ஆன்லைன் ஷாப்பிங் ஆப். இதில் க்ராசரிஸ் எல்லாம் அவைலபிளாக இருக்குது. மற்ற ஆப்ஸோடு கம்பேர் பண்ணும்போது இதில் குவாலிட்டியும் அதிகமாக இருக்கும், அதே மாதிரி டிஸ்கவுண்ட்டும் கிடைக்கும். ஸோ ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோர் ரெண்டுலேயுமே இது அவைலபிளாக இருக்குது. டிஸ்கிரிப்ஷனில் லிங்க்கும் டீட்டெயில்ஸும் இருக்குது. செக் பண்ணிவிட்டு இன்ஸ்டால் பண்ணி பை பண்ணிக்கோங்க. இது ஒரு ரொம்ப யூஸ்ஃபுல்லான ஒரு ஆப். அண்ட் இதில் ஒரு இம்பார்ட்டண்ட் விஷயம் என்னென்னா டோர் ஸ்டெப் டெலிவரியும் அவைலபிளாக இருக்குது. ஸோ மிஸ் பண்ணிடாதீங்க, யூஸ் பண்ணிக்கோங்க. ஸோ டுடே we are going to see about gram staining. First of all, why we have to done gram staining? Because to understand the chemical and theoretical basics of differential staining procedure, here we have to differentiate the gram positive bacteria and the gram negative bacteria based on the stains. Okay. Now, before going to procedure, we have to see the principle. The principle of gram staining is the most important differential stain used in bacteriology is the gram stain, named after Dr. Christian Gram, who invented this gram staining. It divides bacterial cells into two major groups. They are gram positive and gram negative, which makes it an essential tool for classification and differentiation of microorganisms. Those bacteria accept and retain Gram's dye, like Gram's crystal violet, are called gram positive bacteria. If the bacteria loses the Gram's dye, which means crystal violet, are termed as gram negative bacteria because they utilizes the secondary strain, that is counter stain safranin. Okay. Then the gram stain uses 4 different reagents and their mechanisms actions are follows. Crystal violet, it is a primary stain. Crystal violet is a cationic stain. Okay, our bacteria are anionic cell wall, they contains anionic cell wall. So the cationic stain, which is attracted by anionic cell wall and stains all cells purple in color. In appearance, all cells will be purple in color after adding the primary stain. Okay. Then the Gram's iodine, which is a mordant reagent. Mordants in the sense forms an insoluble complex by binding to the primary stain, and this results in the CVI complex, which means crystal violet iodine complex. This serves to identify the color of the stain and all the cells will appear purple black. Okay. But only in gram positive cells this CVI complex binds to the magnesium ribonucleic acid, which is present in the cell wall of gram positive bacteria alone. So it binds to the magnesium ribonucleic acid component of the cell wall. This results magnesium RNA CVI complex, mgRNA CVI complex is formed here. So it is more difficult to remove that the smaller CVI complex. Okay. Then we have to add decolorizer, which means alcohol or ethyl alcohol or acetone, which is 95%. It serves as a protein dehydration agent. Okay. Its action is determined by the lipid concentration in the microbial cell wall. Our bacteria's lipid concentration will determine this decolorizing reaction. In gram positive cells, they contain low lipid concentration. Okay. Then the mgRNA CVI complex will not remove while adding decolorizer. Okay, because small amount of lipid is present in cell wall of gram positives. But by adding alcohol in gram negative, there are more number of lipids content present in the cell wall of the gram negative bacteria, so they are removed by protein dehydration process occurred in the alcohol reactions. Now the cell wall contain a lipid, they are proteins, they are washed out, dehydration occurs there, and there is no purple color or no CVA complex are present there. The cell is empty, stain there. Now this facilitates release of unbound CVA complex, leaving these cells colorless or unstained, that I told earlier. Saffron is the final stain, which is the counter stain, which stains decolorized cells only. Okay, gram negative cells appear in pink in color and gram positive cells appear purple in color. Okay, this is a principle of gram staining. Then now procedure. Initially we have to prepare the thin smear of given bacteria on a clean glass slide and allow the smear to air dry and fixed with heat is must here. Then place the slide on the slide rack for staining, which is present in our laboratory. Then flood the smear with crystal violet and allow it for 30 seconds to 1 minute. Initially the primary staining crystal violet is added, which strains all the culture present in the slide, and we have to wait for 1 minute. Then we have to add mordant, that means grams iodine, which enhance the crystal violet staining by forming CVA complex I told earlier. Then by adding Lugol's iodine or grams iodine, here the binding will stronger. Then we have to kept for 1 minute and wash with running water. Then we have to add the decolorizer, which directly wash the lipid content, so the gram positive cells retain their color but the gram negative cells loses their color and becomes empty in color. Then we have to add the counter stain. This counter stain is a saffronin, or we have to add carbon friction, but mostly saffronin is added in gram staining. This saffronin colors the gram negative cells alone. We have to keep it for 60 seconds and wash in running tap water. Then here we have see the results. Gram positives or appear as purple in color and gram negatives or appear as red in color. Mostly gram negatives or rod and gram positives contains more cocci. So if you understand the video, please like the video and subscribe to my channel. Thank you for watching this video. Subscribe my channel for more videos. Thank you.